Take your next bite. Go ahead, chew it, swallow it. And here's the thing, before that piece of food has even made it to your stomach, a signal has already left your gut and landed in your brainstem. Not a hormone drifting lazily through your bloodstream. An actual electrical impulse, moving down a dedicated nerve, faster than you can even register that you've started eating. Most of us grew up picturing digestion as a one-way street. Food goes in, the gut breaks it down, nutrients get pulled out, and somewhere down the line your brain finally gets the memo that you're full. That picture is just wrong. The gut and the brain are talking to each other constantly, in both directions, and the fastest line in that conversation isn't chemical at all. It's synaptic. It runs through a nerve almost everyone has heard of and almost nobody actually understands, the vagus nerve, and it works less like a mood and more like a reflex. So here's mechanism number 1, and it's the one you can actually feel happening. The vagus nerve is the longest cranial nerve you've got, starting at the brainstem, running down through the neck, through the chest, and branching out across your stomach and intestines. Somewhere between 80 and 90% of its fibers are afferent, meaning they're carrying information upward, from gut to brain, not the other way around. Sit with that ratio for a second. Your gut isn't mostly listening to your brain. Your brain is mostly listening to your gut. Buried in the wall of your stomach and the top of your small intestine are mechanoreceptors that pick up stretch, the actual physical distension caused by food and liquid filling that space, plus chemoreceptors tuned to the chemical fingerprint of whatever you just swallowed. Glucose, fatty acids, amino acids, osmolarity, all of it. Once your stomach wall stretches past a certain point, those receptors start firing. The signal climbs the vagal afferent fibers straight to a structure in your brainstem called the nucleus tractus solitarius, the NTS, which then passes it along to the hypothalamus and other regions that handle appetite, mood, even glucose metabolism. The fastest of these fibers, the myelinated A delta type, conduct at somewhere around 5 to 30 meters per second. Over the distance from stomach to brainstem, that's detectable neural signaling within a fraction of a second of the stretch actually happening. And that's exactly why cutting into the vagus nerve, surgically or with drugs, changes eating behavior so dramatically. Bariatric procedures that disrupt the vagus and the experimental vagal blockade devices tested throughout the 2010s in obesity research both reliably reduce how much people eat. Not because the stomach got smaller, because the brain stopped receiving the stretch signal that normally tells it the stomach is full. Cut the wire, and the meter just keeps filling. Nobody's reading it anymore, but stretch is the blunt instrument here. There's a second mechanism running at the same time, one that's far more precise, and it wasn't even discovered until 2018. For decades, physiologists assumed the cells lining your gut that sense nutrients, the enteroendocrine cells, only ever talk to the nervous system through hormones released into the blood. Slow, diffuse, signaling that plays out over minutes. Then Diego Bojorquez over at Duke University found something that broke that whole model. He and his team identify a subset of these gut cells that form direct physical synaptic connections with vagal nerve endings. Real synapses. The same kind of junction one neuron makes with another. He called them Neuropod cells. In a 2018 paper in Nature Neuroscience, a gut-brain neural circuit for nutrient sensory transduction, Bojorquez's group used a modified rabies virus as a tracer to actually prove that physical connection existed and showed these neuropod cells release glutamate, the same excitatory neurotransmitter your central nervous system runs on, straight onto the endings of vagal afferents. The transmission time they clocked, under 100 milliseconds. Not minutes, not seconds, milliseconds, roughly 3 orders of magnitude faster than the hormonal route. Think about what that actually means. Somewhere in your gut lining right now, there are sensory cells that are functionally an extension of your nervous system, cells that taste your food and report back to your brain at synaptic speed, using the exact same neurotransmitter chemistry as thought itself. Your gut isn't just a tube that digestion happens to occur inside. Part of it is wired like brain tissue, because in a very real sense it is brain tissue, it just ended up somewhere unusual. Now the slower channel, humming along underneath the fast one. As nutrients touch the lining of your small intestine, enteroendocrine cells start releasing a whole cascade of hormones into your blood. Cholecystokinin, CCK, released within minutes once fat and protein hit the duodenum. GLP-1, glucagon-like peptide released by L cells further down the intestine in response to carbs and fats, and peptide YY, which keeps climbing for up to an hour after you eat and helps drive that longer, drawn-out feeling of fullness. None of these need synapses. They travel through the blood to a brainstem structure called the area postrema, a region that sits deliberately outside the blood-brain barrier so it can sample whatever's circulating directly. There's also a hormone that works the opposite way, one that climbs before you eat, not after. Ghrelin, discovered in 1999 by Masayasu Kojima's group in Japan, gets secreted mainly by the stomach and rises steadily in the hours between meals, peaking right around the time you'd normally sit down to eat, then dropping fast within an hour of food hitting your stomach. Ghrelin doesn't just make you hungry, it also acts directly on vagal afferents, sensitizing the very same fibers that stretch will later trigger. It's turning up the gain on the whole circuit before a single bite has even arrived. So you've got a slow wave underneath a fast spike. The Neuropod vagal circuit hands your brain a millisecond snapshot of what just showed up, while the hormonal cascade spends the next 30 to 60 minutes filling in the rest of the picture. How much you actually ate, what was in it, how urgently your body needed it in the first place. And there's a third layer on top of both of those, running on a time scale of hours to years instead of milliseconds to minutes. And this is the layer researchers are still working to fully map. 90% of the serotonin in your entire body isn't made in your brain. It's made in your gut by enterochromaffin cells lining the intestinal wall, something first mapped out in detail by the pharmacologist Michael Gershon at Columbia University, who spent decades documenting what he eventually started calling the second brain, a network of over 500 million neurons packed into the gut wall, more neurons than exist in your entire spinal cord. That gut-made serotonin doesn't cross into your brain directly. It can't get through the blood-brain barrier, but it acts locally on vagal afferents, and those afferents carry the information upward anyway, which means your gut's own serotonin production can shape what your brain hears, without a single molecule of it ever leaving your abdomen. Stack your gut microbiome on top of that, the trillions of bacteria living in your intestines, and you get another layer of signaling molecules entirely. Short-chain fatty acids like butyrate, produced when gut bacteria ferment dietary fiber, have been shown to activate vagal afferent neurons directly. John Cryan and Ted Dinan, at the APC Microbiome Institute in Cork, Ireland, ran a series of experiments, now cited constantly in this field, on germ-free mice, animals raised with zero gut bacteria. They found measurable differences in stress hormone response, anxiety-like behavior, even hippocampal gene expression, compared to mice with a normal microbiome. And here's the part that really nails it down. When they cut the vagus nerve in these animals, several of the behavioral effects of microbiome manipulation just disappeared. The vagus nerve wasn't a side detail in that finding. It was the actual wire the signal was riding on. There's something almost poignant buried in how long it took anyone to notice any of this. When Gershon first started presenting his second brain hypothesis at conferences back in the 1970s and 80s, it was met, mostly, with polite dismissal. The idea that the gut housed an independent, self-regulating nervous network with more neurons than the spinal cord struck a lot of his peers as a stretch, an overreach from someone studying what most people considered a minor organ. It wasn't until his 1998 book, The Second Brain, and the decades of enteric nervous system research that followed, that the field really came around to what he'd been documenting the whole time. Bojorquez's Neuropod discovery, 3 decades after that, was the direct mechanistic proof of exactly what Gershon could only infer from anatomy and behavior back then. An actual synapse, an actual millisecond timestamp, confirming that your gut had been talking to your brain in complete sentences the entire time. Now, the evidence calibration, because these 3 layers sit at very different levels of certainty, and it's worth being honest about that. The vagal stretch response and the Neuropod synaptic circuit are directly measured, with published conduction times and confirmed synaptic anatomy behind them. That's about as solid as gut-brain science gets. The hormonal cascade, CCK, GLP-1, PYY, is decades deep in the literature and already clinically exploited. It's the exact same pathway GLP-1 receptor agonist medications work through. The microbiome vagal connection to mood and behavior is real, and it's been replicated in animal models. But the direct translation to human emotional experience after a single meal is still being worked out. The physics and the wiring here are proven. The full subjective experience they produce in you, moment to moment, that part is still being mapped. So here's what actually happened in the few seconds it took you to swallow that first bite. Mechanoreceptors in your stomach wall started firing as it stretched. Neuropod cells in your intestinal lining, cells wired directly into your nervous system, began releasing glutamate onto vagal nerve terminals within milliseconds of touching your food. That signal was already climbing toward your brainstem before your hormonal system had even started its slower 30-minute build. And underneath all of it, bacteria you will never see, producing molecules you will never taste, were quietly setting the baseline sensitivity of the very nerve carrying the whole conversation. You didn't consciously decide when to feel satisfied. You didn't pick the mood that settled in an hour after eating. Some of that was decided in your gut, by cells doing something that looks a lot like thinking, then reported upward through a nerve that's been quietly running the oldest, fastest conversation in your body. It's worth sitting with the order of operations here, because it flips the story most of us grew up with. We like to picture the brain as the command center, sending instructions downward: hungry, so eat; full, so stop. The actual physiology mostly runs the other way. 80 to 90% of the traffic on that nerve travels up, not down. For a large part of every single day, your brainstem is in a listening posture toward your intestines, not a commanding one. The organ you think of as the seat of decision-making is, at least when it comes to digestion, mostly downstream of a few feet of tissue that most people never think about at all. And that conversation starts all over again the next time you eat.